Well, thank you all for coming. It's a great pleasure for me. So I'm Matthias Wolf, faculty at OIST, and it's a great pleasure for me to introduce my friend and colleague, Professor Mihnea Bostina. Mihnea, just to give a brief overview over his very illustrious career, really. He studied at the University of Bucharest in Romania, where he was a double graduate in both mathematics and geophysics. And after that, he started a PhD in ecology and mathematics at the University of Bucharest. However, he stopped this.
I started two. I gave up one and I started the second one and I gave up the second.
So he went to the Max Planck Institute of Biophysics in Frankfurt with Professor Werner Kühlbrand, who is a staple and a trademark, if you want, in cryo-EM. And he studied membrane proteins with Werner there. And at the end of this, with his PhD, he went to Harvard Medical School, where he joined the lab of Professor Jim Vogel at HMS, studying poliovirus, picornaviruses in general. And later, after the postdoc, so this is where we met, and so we overlapped at the time at this lab. And afterwards, he went to McGill and literally established cryo-EM at McGill. It was one of the first Titan-Crieuses at the time. It says here, the 8th in the world. So this should not be underestimated. These things take a lot of effort, even in particular if an instrument is new. And at the time, cryo-EM was not a mainstream technique. So after McGill, he got a job at University of Otago in 2013, where he became a professor in virology and I guess structural biology. And he focused on the oncolytic virus, Seneca Valley virus, and bacteriophages. And also he established volume electron microscopy there. So volume EM is a slice and view technique where you use a microtome that's inside an SEM. And I think you were one of the first ones to image entire organoids, right? Cancer organoids, essentially. And he teaches virology and microbiology, medical microbiology, structural biology, and supervised more than 20 postgraduate students. He's a doctor honoris causa at his home university, alma mater in Bucharest. And he was the president of the Society for Microscopy in New Zealand twice. And so there are other prizes and accolades that I won't mention. And I'm really honored to have him here at OIST, where he's doing his sabbatical. And I look forward to your talk.
Thank you, Matthias. Thank you so much for this talk, which is 8 months after I joined here. And I started with some slides when I suppose there are at least 4 or 5 people in the audience who already know these pictures and these areas. Definitely Matthias was in half of those. This is our photos from one of the regions in New Zealand, which is called Otago. So it goes from the high mountains where Edmund Hillary was from Dunedin, actually, where he trained to go to Everest and to the sea. And this, we start with this. And this province is here in Otago, and the red is the capital, is in Dunedin. So New Zealand has about 5.2 million people, and Otago population is about 250,000 people. And this is about a quarter of the population in the South Island. And the South Island is much more, has a larger surface area than the North Island. So really, in those photos, if you didn't see any people there, it's true. You can drive for hours and you don't see any car. So that's, just to have an idea how New Zealand looks in comparison with Japan. And as I said, here is a very, those photos were mostly from here or from the seaside. And the University of Otago is in Dunedin, which is in Otago Peninsula. They call it the capital of wildlife of New Zealand. here on the tip of the peninsula is this the only on the continent. Okay, we call New Zealand continent, so we feel a bit more important. Of a colony of royal albatross, which is the largest bird ever. It's like 3 meters when it opens the wings, and I really are my favorite birds. Here is a view from somewhere here, and this is the University of Otago, which was the first university in New Zealand, the oldest, and was a bit later established in Melbourne and Sydney, let's say, so it was 1869. Okay, so that was just a few introductions, just in case some people show up. And this is the title of my talk. And after I send the title, I say, okay, I'm fine with origami. It doesn't mean much, but it's a metaphor, people will come. But after that, when I said, this is a general audience talk, so maybe insights is not the best title I could give. So probably a better title would be structural lesson from infectious diseases, because what I will try to do is to give you an overview of structural virology with the most essential events, chronological, historic events, and also conceptual events. What were the main step forward for structural virology? And if I start with microscopy, because Matthias said I'm a microscopist, I want to start with probably one of my favorite scientists ever, which is Robert Hooke, which is to me is the most complete scientist who ever lived, who in 1665 published this book called Micrographia, which is a very, very important book for several reasons. First, it shows it was a very huge popular success. Yeah, at this stage in the 17th century, everybody read it. Secondly, was probably the beginning of scientific pictures. Yeah, it was really fascinating to see the fly, the tick. And third was a very important book because it was really a multidisciplinary book. It showed us how to do microscope, he showed us how to do experiments, and also he showed results. And finally was a book which showed us that, introduced, for instance, the term of cell. Yeah. It was in this book, it showed that the cork cell also was introduced it. And was probably and that would be the first lesson, that he showed us that structure can tell us something different. It's a whole universe that's smaller than what we can see with the eye, which we can see with the microscope. And less than two decades later, Leeuwenhoek sent a letter to Hooke where he described for the first time a bacteria, and that was the beginning of microbiology. And for like 200 years, of course, people got better and better microscopes. And at the end of 19th century, people started to name bacteria basically on their morphology seen through the light microscope. And now at the same time, in the end of 19th century, people started to discover the first viruses, which were very, very strange biological entity because they were basically described by what they are not. They were not filterable through a 0.22 micron filter. They were not visible with any of those microscopes, which at that time they were quite okay microscopes, and also they were not capable to replicate. People cannot culture them by themselves. So this was the status for about almost half century, till the 30s, when Ernst Ruska developed the first electron microscope. So he was a physicist, and he worked with a lot of engineers. And over the 1930s, there were a lot of new microscopes where, the first one, I think, had a magnification of about 400 times. But maybe not many people know that his brother, Helmut Ruska, was a medical doctor. And his idea was, we can use this microscope to look at all these infectious agents which are actually invisible through the light microscope. And the first images obtained with an electron microscope were images of viruses. And this is a kind of famous, not very impressive, but it was a famous image where you can see an E. coli bacteria with a lot of bacteriophages around it. And over the next, like 10 years, by the early 40s, Helmut and Ernst Ruska published more than 20 reports of different types of virus from different types of diseases, let's say, who were visualized by electron microscope. And that was kind of, at that stage, started to be the gold standard for seeing, you have to see a virus to be able to characterize of an infection. And also at that stage, of course, people started to name the viruses according to how they look in electron microscope, the same like they named bacteria like bacillum or spirochetes or something like that. And we have coronaviruses because they look like a corona. I have the astroviruses, they look like a star. We have toga viruses because are enveloped. We have filoviruses because are a phylum. They have baculoviruses because they have rod. They have rhodo, and we have caudovirales, which they have like a tail. So we'll come back later. So yeah, so the viral nomenclature, and I can tell you a lot of names, which basically is based on electron microscope. This was in 1930s, 1940s. So those images showed us the size of a virus. Also at the same time, people started to get more. So just think that the structure of DNA was discovered in 1953. So where about that time, people started to know a bit of biochemistry, biology, genetics. So they knew the size of the genome of the virus, and they knew the size of a virus. So it was impossible that a virus codes for a protein which is so large as that, as what we see on EM. So people started, okay, the only way to make such a big viral particle is to use multiple protein of the same type. And we're talking mostly about, the viruses which we knew that are icosahedral. We can put an influenza virus and we look like a puddle there, basically. But people put other viruses like polyoma, and they show poliovirus, and they show that they have a kind of isometric image. And Watson and Crick, the Watson and the Crick come like 3 years left after their DNA structure, come with this idea that it has to be a regular polyhedron, yeah, because that's the only way to build a cage with similar protein. And based on this, the only one which made sense because it has the largest volume per the same, the most economic to make with a certain number of capsid protein will be an icosahedron. And this stayed like that until the first X-ray diffraction, not structure of viruses, they shown a 5-fold, a 3-fold, and a 2-fold symmetry axis, and this was clear that is an icosahedron. And that moment, Kaspar and Klug got later a Nobel Prize for developing some algorithm for 3D reconstruction. They come back with this diffraction spot, and a bit later, they kind of made this idea of how we can build a icosahedron capsid from identical protein. And their idea was that you can build very simple, the most simple icosahedron. You just put each protein will be here like a comma, and it stay in the same conformation. However, what's happening if you build larger capsids? So their idea was to introduce this triangulation number, and this is a famous series of articles of Kaspar and Klug in 1962, where they introduce this concept of triangulation, which is not a biological or all new complex, it's a very pure geometrical, which they say, okay, I make hexagons from my protein, and I can make a tiling of a plane, a continuous field of hexagonal 6 slices pizza, each slice is a protein. And I can make from the same protein pentamers, yeah? And those pentamers, they will be a bit smaller. And of course, because you have to close the slices of pizza, they will introduce a curvature. And if you introduce a curvature, replacing a hexagon with a pentamer here, and one here, and one here, you will introduce a curvature. And finally, if you do that regularly enough and not randomly, you will close into a icosahedron. That's geometry. And now, how do you do that? The rule by which you do that will tell you something about the dimension of the hexagon. And this is the triangulation number. And this is, you go H steps on the H axis and K steps on the K axis, and you make this formula, and this gives you the triangulation number. Now, this triangulation number is very important because it tells us several things. First, it tells us how many individual conformation the same protein will have to have in this lattice, yeah? So if this number is 1, like the pure, it's simple. If it's 3, means the same particle will have 3 slightly different conformation in the capsid. And if the number is, if you go 1 and 2 and 1, this will be 7. And if you go 1, 2, and 3 and 1, this will be 13. So that protein will adopt 13 slightly different conformation. That's one thing. And the second thing, it tells if I multiply this asymmetric unit times 60, which is the symmetry of an icosahedron, gives me the total number of major capsid protein existent in that capsid. So yeah, one will have a 60 proteins, a t equals 3 will have 180, and this one you have to do multiplication in your head. So 720, I did it already. Okay, and probably the famous one will be this triangulation 3, where you have pentamers and hexamers, and you can make a icosahedron which looks very much like a soccer, okay, football ball. Okay, and in order to do this geometry with molecules, Kaspar and Klug introduced this molecular switch concept, which means said that each major capsid protein will adapt to an equivalent symmetrical position by moving some very small arms or loops, which will adapt that protein to that specific symmetry. And the idea is that you have one, and how to get this kind of 1,000 sometimes proteins assembled in a single capsid? Simple, you have a protein interacting with another protein, and they change their conformation slightly upon interaction. And now when the third protein comes, it cannot do the same kind of interaction. So it has to do a different interaction, and so on. Now, those different structures, they will be very, very, very similar, except the very, very different position, which will be probably the apex here or some kind of changes in the curvature. So that was the idea of triangulation number. And is that you can use multiple copies, but you can allow for some flexibility. And this will be another lesson of structural viruses. Okay, so this was pure mathematics at this stage. Yeah, no experiment except those X-ray diffraction spots which show that it icosahedron. Now,
sorry, what does flexibility mean here?
Sorry?
What do you mean by flexibility?
I can show you a bit later. So basically, arms like that or loops like that, they would just be slightly flexible in order to be able to make pentamers, hexamers, combine pentamers with hexamers, combine hexamers with hexamers.
So you do not necessarily close your suture.
In order to be able to close that, yeah. So in the 80s, we started to have the first icosahedral structure. Well, a lot of reason why we couldn't have them in the 1950s when the first myoglobin and hemoglobin were solved. We had to wait about like 20 years more. Steve Harrison, one of the second boss of Matthias, described the first virus, icosahedral virus structure in 1978. And a few years later, the poliovirus and rhinovirus were also solved. So this was a structure which had what we call a jelly roll fold, which is basically this kind of beta barrels with 8 strands, which stays basically like a beta sandwich more than a barrel. Poliovirus, they don't look anything. This is a plant virus, tomato bush stunt virus. Yeah, yeah. So this is plant virus. This is a human virus, both polio and rhinovirus. We knew that they are a bit similar, but nothing similar between them. And still, the structure looked exactly the same. You have always not exactly the same, but the same fold, what we call in structural biology. So basically, this jelly roll fold, or this we call it single jelly roll fold, and it has basically has this colored part which is always conserved. So in this case, that was the idea of Kaspar and Klug, that some of those arms and loops, so basically these molecular switches, will be able to adapt in order to make capsids out of these proteins. Yeah, so we know all of this because we've seen them in a structure, and we've seen all this individual conformation in that structure. So poliovirus and rhinovirus were the first, let's say, animal virus ever solved, and they had the same structure. And this structure actually is probably in animal viruses, or maybe in eukaryotic viruses, is the most successful fold ever. We find it in a lot of viruses. This has like hundreds of members of viruses described, and this is what we call in taxonomy an order, and has the most number of families. And one of the families is called picornaviruses. And one of the lessons I want to take from here is that what we see during the evolution conserved is the structure of the protein and not the sequence. Yeah, those viruses, they don't have absolutely no similarity in them, and even viruses in the same families, they may have very, very little similarity. However, when you solve the structure, you realize that the structure is very, very similar. It's like a house. You can replace each piece of brick with a piece of wood, and you'll have a completely different chemical characterization of that house, but the function of that house will be the same. Oops. Just a second, I have an error here on my screen. Okay. So as I said, the picornavirale has many members, many families, and picornaviridae is the kind of the family which gave name to the order, and it has this architecture with this beta barrel or single jelly roll, which they are coming from 3 proteins. So what we've seen before, we've seen that, okay, you have one protein which basically fulfills different equivalent position, but what other viruses are doing, instead of making that protein do some kind of molecular gymnastics, which might be hard to maintain through the evolution, they replace each equivalent position with a different protein. So I'll have a VP1, which will make pentamers around the 5-pole axis, and I'll have the VP2 and VP3 and green and red alternating around the 3-fold axis. And this is the most studied viral family by far. There are over 500 structures deposited in the PDB. They are quite important. Of course, poliovirus, the first animal virus ever, human virus ever described. And all of them, the more they were discovered, the more show that they have the same fold. And this is an old 2019 article, but now tri-EM will be basically this histogram stays more or less the same, but that blue part is now by far like 10 times bigger than the red part. And of course, if you look at the structure, that you have the same the same fold, beta barrel, what it changes are the N and C terminus and these loops, which give those viruses different features, and they look different structurally. They have the same fold, and of course that means they can bind to different receptor, having different tropism. They can be recognized differently by monoclonal, by antibodies, and so on. So that's how the evolution is being driven by changes in these external loops and arms, instead of being while the fold of the protein stays the same. Okay, so we have these viruses which are very, very small. You've seen on the previous slide, they have about 7,000 nucleotide genomes. This is barely minimal to code for about 10 small proteins. So their genome is in a great proportion dedicated to making the capsid, and these proteins are also important because they have to do a lot of things. Think of that. You have a virus, yeah? They have to assemble into a capsid. Now you have that virus, it's ejected by sneezing or whatever, and it has to protect the genome in the environment against pH, against temperature for tens of hundreds of years. Some of those they can resist in the soil. And after that, in the right moment, has to recognize the host. Yeah, has to recognize the cell and bind to a receptor, and has to do a major conformational change in order to release the genome when it should be done, where it should be done. Yeah, and that's what I have a lot of. Okay. When I was in Jim Hogel lab, and we have seen how poliovirus assembles from protomer to pentamer, makes an empty capsid which is filled with genome. After that, it can bind the receptor, and we get this altered capsid which allows the RNA, the single-stranded RNA, to go out, and it's just basically after that ejected to a membrane in the right environment, and you get up with an empty capsid. And this is the other lesson I want to say, that actually you can have the same protein in a virus which have completely different functions. This is true for biology in general, but this is specifically true for viruses in particular. And now I just want to show you for a few slides about one of the direction in our laboratory. And by the way, most of the results I show here today, because I'm like 8 months here, they will be either done here with Matthias or published at least in the last few months. So this is the Seneca Valley virus, which is a picornavirus. It's an animal virus and infects specifically cancer cells. And this is the work which Laura did for the last 10 years, and she works now in Matthias' lab. And we can see that it recognizes specifically cancer cells without infecting any. I will keep the biology to minimum. Yeah, I won't tell you words which don't make sense if you're not a biologist. So you can see that it infects only cancer cells, while the healthy cells are still not infected. And here you have a tumoroid, a spheroid, a 3D model of cancer, and you can see if you put the virus, it kills them. And one of the beauty of this virus, oncolytic viruses in general, and of therapeutic viruses in particular, is that they are self-amplifying drugs. It's enough to put a few particles there, and the organism, the sick organism, will do everything for yourself. You don't have to produce it, you just have to be able to deliver it to the right place. And even more, as I showed here, it will not infect healthy cells. So this is basically an array of viral particles present in one of the infected cells. So we put a virus in here, what cell will die, and we release about 10,000 particles up to which will go and infect different cells. Now, there were several clinical trials done with this virus, but all of them were done before knowing in the 2005-10 without knowing the receptor. Yeah, so we didn't know exactly how to select patients which would be more likely to respond to this type of treatment. So a group in New York discovered, and we had a small part in that, discovered that the receptor of the SVV is the so-called anthrax toxin receptor 1, or also known as the tumor endothelial marker 1. And we were able to see the structure, and we were able to see how the receptor here in pink is able to recognize the capsid. Picornaviruses are always represented with blue, green, and red colors, the 3 proteins. So we can see exactly how this binds, and we can see how the virus is able to discriminate between anthrax toxin receptor 1, which is the virus receptor, from anthrax toxin receptor 2, which is a very, very similar protein, but is present only in all the cells in the body, not only in the cancer cell. Like TEM8, that's why a cancer marker is present in all only in cancers. And we had a lot of studies on that. I don't want to get into detail. Just want to show one general idea about what we actually, will be probably be online like this week or early next week, which we did here with Nadishka, and when he was a postdoc with a former PhD of mine, who was a postdoc with Matthias. And we look at this, how exactly the virus releases, it's called genome uncoating, releases the genome. And we're able to get the several intermediates, some of them showing exactly how the capsid, it's opening, we call the open particle, and releases the genome. We get an empty capsid at the end, which is slightly rotated. You see, it's bigger, and it has this hole here. And we see what we call an altered particle, an A particle at the beginning. And this is very, very interesting, and it's kind of not so important for general biologists, but for picornaviruses scientists is extremely important, because was always a discussion if all the picornaviruses behave like the enteroviruses, which is poliovirus, which they always go through this altered particle, A particle intermediate before the genome release. And that allow us to make a model how the virus binds and goes into some endosome, acidification, changes in the conformational of this interior of this protein, and finally release of the gene. Okay, so this is a short, let's say, recap of what we did in like 15 years on SVV. And now, before I go further, I want to ask you if you recognize what's here, left and right. Exactly. So on the left is they say here living coral because it's in the geology museum, and they call living everything which is not extinct. So it's very living coral. And here is a bacteriophage capsid. And what I like about that, okay, if you work with that, you can see the face of Jesus in a toast. Yeah. So of course, more or less the same, biological explanation is. So, but what I like about this is that all these biological constructs are made from identical units, which are made to make contact with each other, and also they are made that this structure to be functional. So, and also you can see this kind of fractal beauty in both of them, where you start with, in this case, with amino acids, and you go one level higher, and you see secondary structure like alpha helices, and after that you go another one, you see single protein, you see tertiary structure, and after you see quaternary structure, how exactly they make this capsid. Okay, that was the break. Okay, so that's how actually people do, this was a dream like for viruses long time ago. And actually that's how how things looked when I decided to start virology within 1990. And that was the structure of viruses. We had a few of viruses solved by X-ray crystallography, but very, very few. And this is a protein, bacteriodopsin, famous protein. And this is the viruses from the smallest to the largest, yeah. Basically round, beautiful things. I found, basically this review convinced me to do virology after I did membrane protein. And that's how it looked in 2000, I forgot, like 2006 or something, 2016, yeah, I wrote it here. So, and you have those viruses making all this universe from the small viruses here to the huge viruses here. So somehow you can be able to study all of them, because these are also experimentally solved structure, not just alpha folded. And interesting, the more people start to solve structure, the less structure they found. So this is the single jelly roll, which is the most successful fold, I said, in the animal kingdom. And it's another very successful fold, which was solved only in 2000, which is the bacteriophage fold. It's HK97 fold, and this is the most successful fold by orders of magnitude in the bacterial kingdom. And there are other two, which is, another one is the double jelly roll, which looks like this one, but actually they are, imagine that here will be the surface of the capsid. That's why I put it. So some they will be parallel and some will be perpendicular to the surface of the capsid. So that's the other surprise, which we didn't know when we started working as a virologist in structure, is actually the viral structure universe is very, very small. There are very, very few folds, new folds. And everything else is just evolution. Of course, as I said before, this evolution is completely lost. You cannot do like you do normal, you have E. coli and yeast, and you have the same kind of, you can figure it out how the structure is. This is completely unrelated. Yeah, sorry.
When you talk about folding or fold, is there a technical definition for it, or is it just like the geometry of something twisted around?
So the fold is basically the topological arrangement of the secondary structure, which are alpha helices and beta strands. And
what topological phenomena do you use in order to define the fold? Is that homology?
It's structural homology. They look the same. If you look at the 2D, it's kind of, you see the alpha helices, you hit the beta strand. You can predict that based on the sequence, and they look the same. They could look the same, especially for related viruses. And after that, all the things which stick out, basically, they are just
so that red text in the bottom amounts to saying that the virus structure universe has only finitely many configurations of homology.
Yeah, but at this stage, we know there are only four types of. Everything else is basically similar to what we found. And actually, I will show you an example by going now to the most successful fold in the in the life, which is this HK97 fold, which looks like that. You have an axial domain, and this is, you make 6 pieces of that, and you get that kind of 6-fold pizza or 5-fold pizza, and you have a peripheral domain at the periphery of this hexagon, and an E loop and an N-terminal loop. And now that's structure which we solved in my lab, and all of them you can see, you can now realize why do I say they have the same fold? They kind of look the same. Here is one which I still kept it here because it's being processed now, a virus, you know, the Mandarin virus. Okay, these are named in Maori words for some viruses, this was done like a few weeks ago, and now it's being done, and I thought I would be able to put it before the talk. So as you can see, the HK97 fold is very well conserved. What you can have the N-terminal, it can be a bit longer or a bit more curly. You can have this axial domain like a bit more rich, or you can have, in some cases, some inserts in this EF loop. Yeah, you can put here other domains, but otherwise the whole major capsid protein fold is very, very similar. Okay, so how do you do a capsid out of that? Okay, first, it's you don't innovate. If you want to build a new virus starting with an old one, which was the evolution wants to do if you want to go to another host, you don't innovate a new major capsid protein. You just improvise different types of adapters and different types of external morphology, which will give you access to different types of receptor in order to change the host. So how will you build a small icosahedral capsid, which will be like the range of about 40 kilobase per genome? This will be with a triangulation 7, so 7 times 60, 400 major capsid protein. I will come later and I will tell you that this is not exact 420, but let's say for the purpose of a capsid, this is true. So you multiply 7 by 60, and you build an asymmetric unit. You have 6 protein plus 1. So each of those different colors, they have slightly different structures, but the fold is very, very, very similar. And I just will show them in surface because it's easy. And how do you do it? So you put the 5-fold symmetry and you get 12 pentamers at the 12 axes of symmetry, and you put the blue or cyan one, and you put the blue one, and you put the green one, and that color one, and the blue one, and the other color one. Okay, so now you have 420 copies of the same protein, each in a different conformation, and you have basically 7 different positions. And you can see the hexamers here, and you can see the pentamers here. Exactly triangulation, exactly what we kind of knew mathematically from the 50s. Now, sometimes you might need, and you see here white, some additional proteins just in order to give more flexibility, more stability to this very complicated viruses. And what you do if you have those decoration protein in the previous image, they were white and small in the middle. If you have a different virus, you might get this different, we call them, they look like hair clips, and you have the pink and the blue, and they adapt in different position. And what they do, they basically clamp together hexagons in order to give more stability to this, the whole capsid. So that's how you make a capsid for a kind of small viruses. And in sometimes you will have to use different decoration. We call them decoration proteins, and in this case really makes sense. And also you can see that sometimes, but I will get that later, sorry. What do you do if you want to have a slightly bigger capsid? Oh, if you have something bigger capsid, let's say a 50 kilobase per genome, you will have to use, you can still use the same kind of overall geometry with triangulation, 420 major capsid protein, but you might have to use different decoration protein like this green protein, which you can see are more bulky. Okay, what happens if you want to go to a larger genome? And now we are talking about 70 kilobase pair. And in this case, you have to use more major capsid protein. There's no way to deal with the same kind of economical way of building a T equals 7. So you have to do a T equal 9 major capsid, which will give you 540. And you can see now you have decoration proteins, and you have these 9 different proteins in the asymmetric unit showing, again, 9, the same protein, 9 different conformation. And you have a decoration protein, very interesting, which adapts different conformation according to the equivalent position. So this is the same protein here around the 5-fold, like the ones which are staying here around the pseudo 2-fold axis here. Okay, what happens if you get even a larger capsid? So in this case, you know, like 80, close to 90 kilobase, that you will have to again to go higher the triangulation number. So we have to use, I messed up the multiplication here. And again, you have this capsid with triangulation 9. So you go from a 5-fold to another 5-fold through the 3 steps. And I will have this blue decoration protein and the green decoration protein, which some of them, they will cement the hexamers together. So they will stabilize it. They just can be like a clamp. And some of them, they will come here in the middle and probably they will stabilize the hexamer itself. What happens if you want to get even a larger one? You can go to a triangulation number of 13 and use 780 major capsid protein. And in this case, again, you will have to use different decoration protein. And I showed you again that this one, they look very, very similar with the other ones. Now, another things which you have seen maybe more clear is that you make hexamers and you make hexamers here. These are not identical hexamers. And we know that, and we see that this green protein can adapt to this quasi-equivalent position in the center of these hexamers, but cannot do it in the other hexamers. So some are empty, some are full. And we see in the smaller, the same kind of things here, but it has a lower occupancy, which means has a lower affinity for that position than for the other position. Can I go higher? Yeah, you can go to what we call a jumbo phage, and you can make a capsid with 1,620 major capsid protein, and you can make very, very large capsid protein. And now it's another place where the virus innovate, and they don't use decoration protein, but they use something like a decoration protein, which they insert in this EF loop, which I showed you there. So this protein looks very much like the previous one, but it has this huge extra domain here, which basically you can see them, these little twins, they look like, I don't know, like walnuts, decorating the whole capsid. So that's how you can make these huge genomes. And by the way, these sizes are kind of a scale, or to the scale. Is there anything, okay, these are bacteriophages. The jumbo phages are the largest things you can do with HK97/4. They can go much harder than what I showed you, but basically, not much harder, larger, because it's a kind of limit of how much genome can you put in a bacteria in order to get a phage. But you can get even more viruses. And these are the so-called giant viruses, and a huge universe there. And you have seen on the upper corner of that nice poster from the PVRD, this, which we call giant viruses, and we've seen this iridoviruses, which has this kind of not the genome is about the same size of the jumbo viruses, but the capsid is much larger. So this has close to 9,000 units. I don't say capsid protein, and I will show you later why. And has a triangulation number of 147. So it means that each protein will be in 147 equivalent position in order to be able to make that. And why we looked at this virus is that it basically, here is the SVV, let's say the picornaviruses, here will be a jumbo phage, and this is the diameter of the capsid, and this is the genome. And there is a huge universe here, and maybe you heard about these miniviruses, which are actually, you can filter them and you can see it by light microscope. And there is basically a continuous. The good things of that's the smallest of the giants, which you can still visualize by electron microscope, because things here they are completely dark in the microscope, the light, the electrons would be able to go through it. So viruses can be gigantic. So this iridovirus is what they do. They infect larvae, and the larvae become iridescent. And the beauty of that is that they, for several reasons, they are kind of important for biotechnology because they have a lot of interesting properties. So they have a diameter of about 200 nanometers, which is the wavelength of the light. And if they make crystals in the infected larvae, and if you hydrate and dehydrate the capsid, the crystal, it will just change slightly the dimension, and you can go from blue to red. This is a quite interesting system. It's huge. It's huge. It's 1 gigadalton protein. It's a huge on any kind of measure, and that's how the micrograph looks at 1.5 pixel angstrom per pixel. Just okay. And another interesting thing you see here, maybe more here, that you see then why they are so large when they basically have the same genome as a jumbo phage. They are so large because they have a membrane inside, and they are so large because they have a lot of other proteins inside this associated with the genome inside the capsid. So somehow we know that there's membrane inside probably, and there are a lot of other studies which they show that these particles kind of open on one of the 5 folds, and this membrane is able to fuse with the cell membrane and release everything in the cytoplasm. Now, geometrically, this huge particle is also interesting. So I said that has almost 9,000 units because these hexagons are not really hexagons, but they are made of 3 proteins or not of 6 proteins, because the gene which makes them, the protein is actually a double jelly roll fold. And I showed you that, and that's one of the main 4 structural folds in the viral universe. And interestingly, the asymmetric unit, which makes you put that 65, and you get it considered, doesn't have anything to do with the way which the virus assembles itself. So the virus is made of pentasymmetron and trisymmetrons, not of asymmetric units, which is very different from other viruses where usually the asymmetric unit is formed of single protomers which assemble in pentamers and so on. And of course, it's huge, and you will need a lot of decoration protein which will be able they have to cement this capsid to be stable. And when we did that, we also discovered a very interesting particle class which doesn't have pentamers. And when you reconstruct that, you can see that it makes capsids which are made only in these trisymmetrons. Yeah, so you can see here the seams between these trisymmetrons. So you can make capsid with this major capsid protein without starting from the pentamer, which is somehow how we believe everything happens in all the other viruses. Usually have pentamers which act like nucleations for the assembly of the capsid around it. And if you now compare the round empty capsid with the full capsid, you can see that the plaque, actually they have a very, very elastic behavior, and actually behaves like one of those, you get in the proteins here, they just go higher and the other one goes lower. And they make from this spherical structure, which we've seen in the previous slides, this kind of more angular structure, which you have seen in the blue structure, which is the full capsid. Okay, that's how it looks from one side. So it's 10 times bigger, but that's how some major capsid protein hexamers, they go up, and the one in the center, they go down. Okay, so we looked on all these viruses. So this is the last one we talked. This is the picornaviruses. This is a spike from a coronavirus protein. We did a lot of work on that, but I didn't want to talk about this. And these are the bacteriophages. So we know how this releases the genome. We kind of know how this it does because it has a membrane, and that membrane fuses. How does this go? I mean, another thing is that this is a virus which infects a major pest in New Zealand. This is an oncolytic virus. They can be used to kill a lot of pathogenic bacteria. So that's one of the final lessons, is that the viruses can be allies. So they are not always enemies. Okay, so how do those guys do? And now I would need another talk just to give you the next two slides, which are the last slides. Basically, one of this five-fold axis is different. When I tell you that it's 420, they are actually 415 because one of this pentamer is replaced by a structure. Yeah, this actually starts here. The red one is called portal, and it acts as a nucleation for the full capsid, and the capsid forms around that, and these things will have to somehow assist the genome from the capsid in the bacteria. And we know that no protein goes into bacteria. We know that from the 50s when a Nobel Prize was given for that. And we know that the bacterial cell is pretty, I have two membranes, it's a lot of things between the membranes. So it's very, very hard for this small stub to go. So probably the membranes will be more than from here to the floor compared to the size of the phage. So what it does actually, we look, of course, at the major capsid protein and at those two decoration protein, and we can look at the interior of the virus, and we can discover a completely different structure, which is we call ejectosome. And this structure, it's ejected by when the virus finds the receptor, it's ejected, goes through the bacterial membrane, and refolds and makes a tunnel. And that's how those podophages, these short tail phages, are doing this kind of infection from the capsid, which stays outside to the interior of the bacteria, which only the genome is being translocated. How do they do other viruses? So we call this central column. That's another virus, which is biophages. It also has a tail. The tail is much longer. And if we look at it, this was very recently published in the last couple of months. So we have a capsid, you have decoration protein in green, and here, red portal, an adapter protein, and here you have a connection between the tail and the portal, and at the other side, we have this base plate. Yeah. So this base plate is responsible for finding the receptor and somehow triggering the collapse of that helical structure in order to release a protein, which is here in yellow, which will go and penetrate through the double membrane of the bacteria. And that's kind of how many proteins are viruses like this one and this one using just for that, let's say, cell puncture apparatus. And this is another lesson. We've seen that simplicity is a winning strategy. We've seen that with single jelly roll fold, and that with the other lesson will be that complexity is a winning strategy because those viruses, they devote up to 40% of their genome just to make a white capsid. And to give you an example how amazing they are, you need one phage to kill one bacteria. You need at least 200 particles of poliovirus to kill one cell. So these are cost a lot to make one, but they are perfect. Okay, so the final lessons, and here all the other lessons which I showed you before. And of course, I want to thank my lab, and I want to thank Matthias, who basically most of the structure we use here were done with his microscopes, with a microscope from OIST, and I have Mike and Jay from Jay was also here in Matthias' lab. We did some other viruses. Rafa, who wrote some of the scripts to do this beautiful structure, Nadishka. I wanted to ask Anna a photo, but I said, no, I should better get one from the local newspaper. And these are Laura and some of the students. And actually, maybe you've seen, this is my lab, and maybe you've seen that they like to make very nice figures. So I said that all the figures are made by them. Yeah? So I said, maybe I should do at least a nice legend for this figure. And I just, yeah, I didn't know, but because this was organized by the university, so okay, let's everybody put stripes. And that's how you can recognize each one based on the stripes. So I just want to thank few of them specifically. James, who solved several of those structures. Georgia, who works on this virus, that's a very fresh virus. It's probably like 3 weeks structure, which we started working together with another diploma student in the lab. That's Alice, who solved this virus, and Anne, who solved this virus. and Rosheni, who did all the SVV work and also this virus. And of course, that's all. I think we have 5 minutes for questions, sorry.
Thanks, Mihnea, for this inspiring and awesome talk. Really enjoyed it. So if you have questions, we should use the microphone for the audience in online.
Thank you very much for such a great talk. I have a question about these decorations, additional proteins. When they bind to the main capsid proteins, the structure is a bit different between them, right? Or they are exactly the same?
Yes, actually the easiest is to see with this one, which actually they don't use the same protein because the structure is slight difference, and the blue and you see they are almost identical, but they are actually a bit different just in order to be able to recognize this equivalent symmetrical position here and here. So that's, yeah, they are, some of them, they have arms and they can adapt, and some they are different proteins.
I see. The second question. When you say about the evolution and the structure is more conserved than the sequence, but we have such different viruses, but with the same structure. Is it really evolution, or it's some physical restrictions in nature?
I don't think it's. I think it's basically we lost that track of that evolution. I don't think we can. It's kind of Heisenberg equivalent of evolution. I don't think we will have intermediates from that. And I think it's just like I said. You don't innovate if you can improvise. And it's no way. Probably with all this artificial intelligence, probably we can make a virus from zero. But the evolution is you don't have to make things better, just to make them a bit better for the next infection. Yeah. So I don't think I think on a different planet probably are much different and more viral fold. There's not an attractor in the fold field because the proteins, the cell proteins, they are how many folds are hundreds of different folds. Yeah. So it's no reason to be only 4 because they are viruses.
Thank you.
Thank you. Super cool. When the virus delivers the genome, it reassembles as an empty capsid, right?
Yes.
And can these empty capsids be recycled? Can they grab DNA again and redeliver it? Or is it just not a particular function?
First, you just get an empty capsid inside the virus, so it's nothing to recycle. Yeah, you don't need many viruses to infect the cell. So that's recycling. That is not the point. However, you get empty capsids when you start to assemble. They look different than these ones. They are called procapsids, and those ones disassemble, and basically is a way of keeping protomers in the area where you need them, and they can disassemble and get the DNA assembled again. So that in this way, this empty capsid, not the ones which I showed, they are able to reassemble around the genome. And also it's a lot of work in doing vaccines like VLP vaccines, and just and the easiest way to do it in plants is no way you can replace yeast or E. coli what you can do with plants. In 6 weeks you get kilograms of proteins, and you always get RNA from the cells. Yeah, so they definitely are able to get any kind of RNA, but somehow specifically they get a viral RNA.
Anyone else?
I will shut up. I'm just wondering, you said that, oh, yeah, thank you. You said that, in the capsid form, sometimes one protein can be in different conformation to form a capsid. So I was wondering, when is it decided which conformation that particle molecule will make? Like, is it just based on allostery that, like, based on protein-protein interaction, or it's decided that in the translation or done in the folding?
So the official theory is that once two proteins bind, they will change the conformation, and the third protein cannot bind in the same way. So it's somehow a cascade effect that the third one has to come in a different. So that's the theory. Now, we don't have, even with cryo-EM at this stage, we don't have access to that type of data because these proteins usually are kind of unfolded and they fold and they're usually very small. So that's one of the big questions. If we can get one of those assembly line for one of usually a plant virus or a picornaviruses from single protein to pentamers, that would be a big question. But usually some, especially the more complicated one, they do it separately. So you can get hexamers and pentamers. And the affinity to make a hexamer is like X times bigger than making pentamers. And this is given by the triangulation. If you want to make, I don't know, a T equal 27 with tons of hexamers and just a few of pentamers. If you want a T equal 7, you just have to be a bit better at making hexamers than pentamers. Now, there are phages which they get rid of that problem and they make the pentamers from a completely different protein, which is very similar, the same fold, the same, just to not get into this kind of sequential affinity, or I don't know how to call it, in order to be able to build the capsid according to the affinity between proteins.
Is it possible that chaperone proteins of the host can somehow like control this mechanism?
The virus has its own they has scaffolding protein, and they are the ones which regulate the formation of that. And actually, that's one of the reasons they all look the same in the structure, but they are a lot of procapsid, which is a smaller capsid, which is not so stable, which is stamped with DNA. And the structure in these procapsids, it's very different. So the hexamers are not hexamers. They are very elongated hexamers. And that kind of arrangement, now, if you want to make it with ovoids, not with spheres, it will be much easier to figure out how to do a capsid than if you make with like this kind of tiling, which I showed.
All right, just like the phage, perfect timing. Thank you very much, Mihnea.